All right, are we on? Welcome, everybody. Are we good broadcasting? All right, thanks everyone for coming to our 2024 Chemistry Nobel Prize Colloquium. Usually we do this in a smaller room, and it's the faculty telling interested parties sort of why a general science or chemistry audience should care about the Nobel Prize. So this one's a little special because I don't have to do all the work. We have an actual expert in the audience who's going to take care of us in a few minutes. But what we're going to do first is I'm going to try to take a big step way back and provide some context for what this prize is all about, how it's rooted in chemistry going back over 100 years. And then we're going to move into more recent stuff that David's going to tell us about, if my computer will behave. I lecture in this classroom, and every day my computer tries to mess with me. Usually it's a Windows machine, so I blame Microsoft, but this is the Mac, so thanks a lot, Apple. Anyway, all right, so let's, again, take a big step back to get everybody oriented. I want to talk about what proteins are and why they're important. And most of you know this, but some of you may not. So I want to go back to the central dogma of molecular biology, where the essential biomolecules are DNA that encodes genetic information, transmits that information to RNA, which is another nucleic acid, and that information gets turned into a protein, which is a polymer of amino acids. And then that polymer of amino acids folds into some globular structure. And the really critical thing that's going to underlie everything we talk about today is that proteins are molecular machines that perform biochemical functions, and those functions are determined by their shape and structure. And there's a huge range of, seriously, this computer's, I shouldn't have made a crack about Apple because now it's really messing with me. Okay, now it works again. It's going to stop working in 2 seconds. Okay, so here are just a short list of the many things that proteins can do. So they do things like chemical reactions, they build cellular structures, they do motor-like walks along the cytoskeleton, they mediate cell signaling, control gene expression, do things like transport oxygen around. And the central idea that you want to keep in your head all the way through this is that biological information is encoded as a sequence in a polymer, and that sequence defines a molecular structure in 3 dimensions, and that molecular structure in 3 dimensions defines the function. So the fundamental questions that have been animating the field of protein science and biochemistry for a very, very long time are, if we know the sequences, can we then predict the 3-dimensional protein structure? If we know something about the 3-dimensional protein structure, then we may learn something about the function. And that leads us to our next question, which again is hopefully going to be the heart of what David talks about. If we understand protein structure well enough and can predict structures, can we also design structures? And in particular, can we design structures to perform functions? All right, so that takes us to the Nobel Prize, which you all are aware was awarded to David Baker and 2 scientists at Google DeepMind, Demis Hassabis and John Jumper. David's portion is for the second question, figuring out how to design proteins with well-defined structures and functions. And the other side from Google DeepMind is a huge advance in the protein folding problem, being able to predict protein structures from sequence using deep learning methods. I've added some of the key papers right here. So David's work in protein design, at least from the outsider's perspective, really jump-started with this 2003 science paper. He might go into some detail on that, but if not, go read it yourself. And then the protein structure prediction was a paper that sort of blew up on the scene just a couple years ago in 2021, announcing the release of AlphaFold. Many of you may have used it. You can access it in various new iterations online. Okay, so we're going to take a deep dive and try to explain what's going on with protein structure. But before we do that, I want to get a couple diverse perspectives on what proteins are and why they're important. And so this is a picture of David frozen in a moment in the press conference the day the Nobel Prize was announced, when somebody in the audience asked him essentially, what is a protein? Now, I'm not sure that's exactly the question. We were all watching this on YouTube and we couldn't hear the audience feed. So we don't know who asked that question. I have a sneaking suspicion that it was one of the physical chemists and not one of the organic chemists. But maybe it was an inorganic chemist. Physical chemists can be off the hook. Anyway, so now David had an answer for this question. However, David was possibly sleep deprived when he gave this answer. So David's answer, and I quote, was the proteins are the miniature machines that carry out all of the important jobs in our bodies and in all living things. And you know, this definitely puts a lot of biologists on edge, right? So really all of the important jobs, So, I asked around, I have some friends in this area and asked, what's your perspective? How do we feel about that quote? And I got one immediate response that is that proteins do all the work. That's okay, but RNA is what does all the managing. This quote actually comes from our friend Riju Das, who is a former Baker Lab postdoc, current Stanford professor, and forever RNA partisan. I tried to find a picture of Riju and David together to show just how tight they were, but this is as close as I got them. I asked Riju for a picture of him with David. He came up empty, so this is what we got. But anyway, yeah, if you come up right to the front of the room and look, that's definitely Riju and that's definitely David over there. Anyway, if you care about RNA, it turns out that was the 2024 Nobel Prize in Physiology or Medicine. That and many other Nobel Prizes have been all about RNA and its functional role in doing all sorts of important things. Unfortunately, the carbohydrate and lipid chemists are out in the cold here. So we're just talking about proteins and nucleic acids. We'll come back to that later. But anyway, we're here today for chemistry. So now what I want to do again is take a kind of a historical perspective and set the stage for how we understand what the problem is, and then how did we get to where we are now. And it actually turns out a nice way to do that is to go back through all the Nobel Prizes in chemistry that have been related to protein structure and function, going all the way back to 1946. So we're going to work our way back through a little bit of the history here. So the earliest questions here actually goes back to, what is actually doing all the work? So imagine it's the late 1800s and early 1900s. You've got scientists messing around, taking biological extracts and trying to figure out what biology is actually doing. And at the time, people found out that biological materials, things they purified from living systems, can accelerate chemical reactions. And this is a huge deal. This is one of the early model reactions, breaking down urea into ammonia and carbon dioxide. And so the hypothesis is that there's some sort of molecular species, they'll call it an enzyme, that does this breakdown, that makes this reaction go fast. And people spend many, many years arguing about what is the molecular species that can do this. No, totally not. That's an inorganic chemist answer right there. Anyway, well, okay, all right. So there's probably some metal ions involved, but that's not the immediate question right here. So the 1946 prize was given for a series of, very beautiful chemistry experiments. So starting from back in the day, you don't get your DNA from IDT and just make your biological material. Instead, you actually go out into nature or you buy it from a food processing company, purify from the actual organism, in this case, jack beans, whatever they are. Apparently, it's animal feed. And then purify it. So classical chemistry going all the way through to crystallization to get the most pure possible thing you can get. And then in 1926, actually, so not really 1946, this paper was published saying that the stuff in these crystals, the stuff that has the activity to do this chemical transformation, is a protein. And doing all sorts of classical chemical tests to say the composition of this thing is protein. And very specifically in this paper, they made a big deal about saying it is not a carbohydrate. So it was very important for the chemists of the 1920s to say that carbohydrates are not that important. They're going to feel bad about that when DNA comes along, and then when Carolyn Bertozzi gets the Nobel Prize a couple decades later. But anyway, so again, really big advance, and it seems, things that we take for granted at the time, but at the very beginning of sort of modern science, we don't know what is actually doing the work. So it turns out that protein is doing the catalytic work, maybe with some help from metal ions, but it's really the proteins putting it everywhere. All right, so next big question, or next big step that was recognized. Oh, sorry, I did want to add one point, which is note the paper was published in 1926. The prize was 1946. There were about 20 years of people arguing about, well, we don't really think it's a protein. Proteins can't possibly do all the cool things that you claim they're doing. So this was, at the time this was published and for many years after, really controversial. Things we take for granted now turned out to be really hard to establish rigorously. Okay. So we move to 1954, which was a Nobel Prize to Linus Pauling. And this, why will this little box not go away? Let's try down here. Okay. 1954, one of the key insights that was recognized in the Nobel Prize was that proteins have structures that are really important for function. I don't know what's going on over there. No. Anyway. So this is, this prize was for Linus Pauling. Really, it's the Linus Pauling Lifetime Achievement Award for fundamental advances in chemical structure and bonding. But two things he did in the course of this huge career that's, deeply influential for everything we talk about today. There were two critical advances for protein science that I want to highlight. And one is that my computer stopped responding again. Okay. One is that proteins have defined structural motifs, and this was taking known information about bond lengths and angles and hydrogen bonding patterns and predicting that chains of amino acids, the polymers that make up, or the chemical entities that make up proteins, should adopt well-defined structural motifs. They should fold up into helices, they should pack together in sheets. And again, this is a huge advance at a time when we didn't actually have experimental data on what proteins look like at atomic resolution. So it's a big deal to say, okay, this material, this protein, the stuff that we think is doing functional stuff in biology has a clear structure. And then the other advance that Pauling really formalized, building on work from a lot of other people, was the idea that shape determines function. In this particular case, the example is an enzyme positioning chemical functional groups in just the right place to make a chemical reaction go faster. But really generalizing the concept to antibody recognition, shape complementarity, and so molecules interact with each other by matching the shapes, matching the chemical and physical properties to form complementary interacting surfaces. Again, this is a huge deal, so this is 1954. All right, now we're moving to 1958. I think something's supposed to happen here. No? No? Okay, all right. Anyway, so what actually happened in 1958? So again, we know proteins are the machines that are doing important biological functions. We know their shape matters, but we really still don't know a lot about what they are and what their defined chemical structure is. Even at the time, people are arguing, is a protein sort of an amorphous mix of different molecular species, or is it a chain with a well-defined polymeric composition? And so this takes us to the work of Fred Sanger, who published a paper, losing all my, how do I make this thing go away? Who published some papers in 1946 to 1955, determining what the sequence was of insulin. And again, I want you to set yourself back to the mid-1900s. DNA structure has just been determined in 1953, but we don't know the genetic code. We don't have reliable DNA sequencing. So things we take for granted now, like you know the sequence of an organism and you can predict pretty much all the proteins and exactly what their sequence are, that information we don't have. So at the time, figuring out that a protein has a defined sequence and that sequence is important for its function was really painstaking chemistry in multiple steps over many years. First, just figuring out the big step was what is the residue on the N-terminus of this two-chain protein, and then chopping it up into small pieces and then carefully figuring out how to purify and separate those pieces, and then doing sequential cleavage, one amino acid at a time, and getting about 5 or 6 amino acids at a time, and then fitting the jigsaw puzzle back together to finally figure out exactly what this protein sequence is. So 10 years of work for something that we can do trivially now just by looking at DNA sequence, but a huge advance for the field. All right, now we're moving on to 1962. So we have sequence, we have function, we know the shape is important. Uh-oh. But seriously, me? I already have long hair. Okay, we're in the 1960s. Right, so, but they really did the work in the 1950s. Is this accurate? What would I look like if oh no. Oh, this is awful. Seriously? Wait a minute. Okay. I'm not sure we got this right because this is, we haven't gotten to this area yet. Anyway, all right, so I can't even see my computer anymore. So so what's the question now? Can we determine the structure of a protein? And again, this is challenging. Chemists knew how to determine molecular structures and could do small molecule structures with tools like crystallography, but applying this large molecular species that's a protein is a big challenge. So the prize in 1962 was awarded to Mats Perutz and others for determining the structure of hemoglobin. And again, I want to highlight sort of what life was like for structural biologists in the 1960s. This is a physical model that they took a picture of and stuck in their paper of what they thought the protein looked like at about 5 angstrom resolution. And so the central conclusion here is it's a big old globular blob. Now, their conclusion was a little bit more sophisticated than that, but that's really important. This is the picture that emerged about 10 years later. We actually recognized those secondary structure elements, the defined alpha helices and beta sheets, and all those things packing together to form a globular structure that positions molecular chemical elements to do important biological functions. This one is hemoglobin, so its function is to hold a cofactor in place that binds oxygen and then move it around the body. All right, we're moving on. We're getting close to the point where you're going to hear from David and not me anymore. Okay, we're in 1970s. I get to get rid of this. I was told that you were going to wear the costumes. Oh no. You got it. You're kidding me. Oh, I can't even, you can't even hear me. No, you have to wear this one while I talk. Ah, all right. I'm not a crook. Okay. Anyway, 1972. But again, the work was really done in 1961. So you're off again. Okay. All right. So protein sequence determines structure. The prize in 1972 was, so again, we've got sequence. We know that proteins fold up in structures. How do they go from that linear sequence to a three-dimensional structure? This was a beautiful series of experiments where you take a globular folded protein and unfold it with denaturing agents, so make it a big, long linear polymer, and then show that you can take those denaturing agents away and that the protein spontaneously refolds when it's in a purified state. And what that means is, it's contained in this quote up here, is that the information to determine the globular 3D structure of a protein is contained in the amino acid sequence itself. And again, this is a huge conceptual breakthrough at the time, but it also raises some big questions that are going to set the stage for where we're going now, which is that the space of possible sequences is astronomically vast. So you have a 200 amino acid protein with 20 possible residues at each position. That gives you 20 to the 200th possible sequences. So just a huge number of possible sequences, all of which could adopt, presumably, well, some of them adopt structures, some of them don't. And then the other challenge is the space of potential conformations is just huge. So astronomically vast sequence space, and make that even bigger for the configurational space. And so here, this is commonly framed as Leventhal's paradox, which is that, for a sort of simple 100 residue protein, we can estimate there's maybe 10 to the 70th possible configurations. If those configurations are sampled on the order of once every 10 to the minus 11 seconds, then it still takes 10 to the 52nd years to search that space. And this is clearly bogus because proteins fold in milliseconds. So physics has solved this problem. And there's a lot of work going on in figuring out why this paradox is maybe vast oversimplification of the problem, and that there's energetic interactions that vastly simplify the space very quickly. So decades of work have gone on here, but it also sort of sets the stage for our challenge now, which is, if we know the sequence of a protein, how do we explore that space, figure out what the structure is, and how do we take new sequences and figuring out how we can predict what they might fold into? All right. So that was the 1970s, and then we go to now. So goodbye, Nixon. We're done. Totally. Okay, wait, do you have any more costumes? Are you sure? Okay. I wouldn't have been able to do the hula hoop anyway. Okay. So, all right. So from the 1970s to now, to set the stage for where we're at right now, so there was the big gap in protein science Nobel Prizes, but an equally critical advance happened, which was scientists all over the world worked to study and understand protein structure and function. And as part of that, depositing structures into the protein data bank. So determining experimental three-dimensional structures, depositing them into a database that just sort of blows up. The blue lines, you can't quite see the numbers here, but here in 2021, there's about 200,000 entries. Now, some of us, myself included, have deposited multiple entries of the same thing with minor amino acid variation. So that number is totally unfair. But there's really on the order of 100,000 unique entries that show you different things about protein structure. And that is quite a big number that sets the stage for applying modern deep learning tools to this protein folding problem. Now, this is a chemistry audience, and I'm a chemistry professor speaking to you. So if I actually try to explain what's going on in the machine learning, I'm just going to embarrass myself. So I'm going to turn it over to Mike Gelb right now. No, you weren't ready for that? Okay, never mind. Anyway, so I'm going to tell you from the outsider's perspective what happened in 2020, which was AlphaFold emerged on the scene and literally rampaged through the 14th CASP competition. So CASP is this competition that's been going on for many years, called the Critical Assessment of Techniques for Protein Structure Prediction. And it's a blind test where people who've recently determined experimental structures hold them back for a few months before release and let the computational people try to predict what the 3D structure is and then match them against true experimental data that they've never seen before. And this has been decades of creative failures until AlphaFold came along and really blew everyone's mind by predicting 87 of the 92 possible sequences with really high accuracy. Now, what's going on under the hood of AlphaFold is taking advantage of this huge data bank of protein structures, using deep learning techniques to generate some sort of inferences about what protein structure looks like. And then if you give it an input sequence, it's going to make a prediction. And what that looks like, if you go to their paper, is some complicated diagram of layers and things that I don't understand. I stared at this paper for a while. The key, again, from a sort of distilled chemistry perspective, there's kind of two critical things going on here. One is building a multiple sequence alignment, and then building a representation called a pairwise representation that tells you something about the distance relationships between the proteins, using the multiple sequence alignment and that pairwise representation to try to iteratively refine both of those things and figure out what's the sequence relationship between your particular input sequence and all the other sequences that are out there. And then what is the likely relationship between positions in that structure in 3D space? And then from that, these sort of abstracted versions of what a structure looks like, so sequence comparisons and a matrix of sort of pairwise interactions, take that to a module called the structure module that tries to actually map that information onto a 3D model of a protein structure and iteratively build a 3D backbone, and then at the very end of that, predict side chain geometry and come up with a protein structure. So what are the takeaways from this? Because clearly I don't know what I'm talking about right now. The takeaways from this are that AlphaFold in practice does an amazing job of predicting protein structure and becomes an incredibly useful tool for users like me who want to know what a particular sequence might look like. In the time since 2020, when this came on the scene, there have been multiple different versions. The newer versions use completely new techniques, recently like diffusion, which has been a central feature of some of the work that David's going to tell us about right now. So without further ado, we're now going to switch gears and let David tell us about some recent, his insights onto protein design and how this all comes together. So thank you, and we'll hand the floor over to David.
Okay, all right. Yeah, that was pretty amazing. In fact, this whole new side of Jesse, that was great. And can I borrow that next month? Okay, that was that was really great. No, it's also a hard act to follow. So I'll try and I'll try and stay kind of like kind of big picture, high level for the first part, and then I wanted to get into stuff that I'm kind of excited about now, where I thought that kind of involves some chemistry that I thought maybe some people here might be interested in collaborating on. So like Jesse said, we've been working for a long time on actually first on protein structure prediction and then on protein design. And the way we did it initially was, and actually for many years, was to try and model all the interactions between all the atoms in a protein as accurately as possible. And so to predict the protein structure, we knew the amino acid sequence because of the advances that Jesse described. We also knew that the amino acid sequence contained all the information because of Anfinsen's experiments, which Jesse described. So so what we tried, and another way of sort of restating Anfinsen's results was that the sequence determines the structure, and the most obvious way that could be is that proteins fold to the lowest energy state for their amino acid sequence. And so each it's kind of like a ball rolling downhill. It'll eventually end up in the lowest elevation point. And in the same way, a protein, as it folds up, it makes all these interactions, the energy goes down, and it ends up in its lowest energy state. And so if you want to predict protein structure in that picture, you have to be able to sample different possible protein conformations, and there's a lot of them, like Jesse said, so that was a little complicated. And then you also have to be able to compute the energies of all the interactions between all the different atoms in a protein, and since there are a lot of them, that was also a little complicated. So we developed a computer program called Rosetta over the years, first to try and predict protein structure, and then once we made some progress in going from amino acid sequence to protein three-dimensional structure, we realized we could go backwards and make up a brand new structure and then find a sequence which would be predicted to fold to it. And like Jesse said, many years ago in 2003, we showed that you could actually make up a completely new protein structure on the computer, find a sequence that encoded that was predicted to find a sequence for which that structure was its lowest energy state, and then we made a synthetic gene that encoded that protein. So we're admitting, I have to admit, DNA does something too. You didn't really mention DNA, but DNA is just, it's kind of like, it's just storage medium. But anyway, so so we had to make a synthetic gene because it was a brand new protein with a new sequence. So there wasn't any gene in nature which actually encoded that protein. And then we put the gene into bacteria, and the bacteria make the protein, and we got the protein out, and then we were able to solve the crystal structure of that protein. Actually, Gautam Dantas, who was a graduate student in my lab and working with Barry Stoddard, solved the structure at the Hutch, and it matched the design model. So that was pretty cool. That was like 20 years ago or more. And so then since that time, so we could make a brand new protein structure, but it was like the most boring protein in the world. So it was the only protein that was known to anyone, which really did absolutely nothing. Like, because all the proteins in nature have jobs, but this was a poor jobless protein. It was completely unemployed. So then we've spent ever since then, we've been trying to make proteins that do things. And along the time, there was a lot of ridicule, particularly from chemists when we were trying to make catalysts. We're saying that these are the most pathetic enzymes, or these proteins really don't do their jobs well at all. But we kind of kept going on, and so we got better at making proteins that actually do things like bind to other proteins or catalyze chemical reactions. And the further we got, the more interesting it got, because if you look in nature, like there was that famous quote about proteins doing everything. Well, like there are a lot of different things that go on in biology, and I'll come back to this later. There's we have tooth and bone and stuff where proteins are interacting with inorganic mineral compounds and making these kind of amazing hybrid materials, and proteins capture solar energy, and they do chemistry with it. So there are all these amazing things that proteins do. So over the years, we started just trying to explore, all kind of inspired by what all these kind of crazy things that nature can do with proteins. We thought, well, maybe those just happen to happen. They kind of evolve by random mutation and selection over billions of years. What if we could actually intentionally design proteins to do something? And as we got better at it, it got more and more interesting, because you could think, well, maybe we could actually solve some current problems that face humans where a protein could fix it, but we don't want to wait 100 million years for the right protein to evolve. So like the most obvious places in medicine where you could imagine making a new protein that would be a better drug for treating cancer or autoimmunity, or maybe combating viruses. So we've actually designed a number of proteins for those different areas, and some of them have advanced into human trials. And there's actually now an approved medicine that's a designed protein that's a COVID vaccine made by my colleague Neil King here at the UW. And so we're starting to see proteins and design proteins starting to contribute in medicine. And now, it seems like almost every day I get an email about some new company starting up that's going to be designing proteins to cure cancer or something. So it's become quite a fad, which is kind of interesting for me, because I was kind of on the lunatic fringe for many years, and now it's like establishment. It's kind of weird. So let's see. So, but what happened about what happened that Jesse talked about, in 2020 was the DeepMind showing with AlphaFold that deep learning could be really powerfully applied to protein structure prediction. So that was really exciting when that happened, and it was clear that if you could use deep learning for protein structure prediction, you could also use it for protein design. And so at that point, we just kind of dove right in, and I didn't really know what deep learning was, but we had to figure it out. And so it was really fun. We kind of had to get up to speed quickly, and we first actually what happened at that point, it wasn't really clear what DeepMind was going to do with. Were they going to write a paper on AlphaFold? Were they going to release the code? It just wasn't clear. So we tried to. They had given a talk outlining the ideas at that CAS meeting that Jesse mentioned. So we thought, well, okay, we better try and figure this out. So we came up with something called RosettaFold, which wasn't quite as good as AlphaFold, but it was far, far better than anything else we'd had before. And then, luckily for the world, DeepMind did release their code, and so that's had really big impact. And then we, at that point, dove into trying to apply deep learning for protein design. And so that's where I'm going to begin today. So like if you want to solve a problem, like a medical problem or a sustainability problem with protein design, you first need to decide what that problem is. I mean, that sounds kind of obvious, but it's actually one of the most important parts, because you have to pick a problem which is an important unsolved problem, but where it can actually be solved by protein design, or at least there's a good chance. Well, so once you pick that problem, the first step is to generate a new protein shape that is predicted to solve that problem. Like maybe it's perfectly complementary to a target on a cancer cell, or perfectly complementary to a small molecule you want to detect. And then we have another, and this is done with a program called RFdiffusion, which I'll describe in a moment. Then we have a second program that will take this backbone and now find a sequence which will fold to this structure. It'll direct the folding like Anfinsen showed for his protein, and then the proteins that he was studying. And then not only that, but it will also have the function that you want, like it'll have a binding interface for the target. And then we use either the program we develop, or actually more often AlphaFold because it's better, to predict the structure, the sequence that we have here. And obviously for self-consistency, this sequence should be predicted to adopt this structure. And if that's true, then we again make a synthetic gene that encodes that protein, and we go to the lab and make it, and then we can test to see whether it does what it's supposed to do. So I can give you a little bit of an idea about how RFdiffusion works. And really the key is all that work that happened in the gap that Jesse mentioned, where structural biologists were solving 200,000 crystal structures. And so that's what really made AlphaFold possible, and it's what made RFdiffusion possible too. So first I'll tell you about image generation methods like DALL-E, which probably many of you are familiar with. So the way these methods work is you give it like a prompt, like a cat sitting on a giraffe, and then it makes an image that looks like that. So the way that's done is it's actually done with diffusion models. The way those work is you take all the images off the internet, you add noise to them progressively, and then you train a neural network to remove the noise. Obviously, that gets harder and harder the more noise it is, but the network just does the best job it can. So once you've trained that model, you can start with completely random noise and then progressively denoise it. And amazingly, what you almost always get out is something that looks like an image from the internet, but actually it's a completely new image. And you can train the network to be influenced by conditions that you put on it. For example, a cat sitting on a giraffe. And then this diffusion process sort of goes towards something that looks like, maybe in this case, a cat sitting on a table. And so we did exactly the same thing with proteins. So we took all the protein structures in the protein structure data bank, we noised them to different extents, and then we trained a network to remove that noise. And then once we had done that, we could generate a completely random distribution of residues and progressively denoise it with the network. And what we ended up with were proteins that looked like perfectly viable proteins, but they were completely new. They weren't exactly identical to any protein that was previously known. So that, again, is not very interesting because that's just a new protein structure. That's like top 7. The interesting part is we can condition it in the same way that you can condition the image generation with text. You can condition this process. And here, I'll just give you an example of how you would make a binder using this process. So this is a protein target, the structure of a protein. It's the insulin receptor. It doesn't really matter what it is. But we start with this completely random cloud of residues here. And this whole removing the noise process is happening in the presence of the insulin receptor. And the network has learned how proteins look that bind to other proteins. And it knows that whatever's binding to this one should be very complementary in shape. And you can see out of this noise is coming something that's has a shape that really perfectly matches the target. So we've used this approach to make binding proteins for over 200 different protein targets now, and it's actually kind of funny. Some of the more recent things we've been trying to make designs to things like ion channels, which are really attractive drug targets, or the immune complexes that are on surface of cells, MHC bound to peptides. We've made binders, and then there's a limit to the characterization we can do in my lab, and so we have tried to contacted people who are experts in the world to see whether they'd be interested in testing them and collaborating them. And the response we've gotten the last month several times now, actually both the ion channel and the MHC binder cases, yes, we'd be happy to collaborate. And by the way, we've been using your methods to make our own binders, and we already have some. So it's really kind of cool. So lots of people are using these methods, and so we can make binders to things like the amyloid fibrils, that A beta that are in Alzheimer's. We can make binders to viruses that block the viruses from infecting cells. We can make binders that look very promising in cancer models, and a lot of things like that. So, but what I thought I'd do today is talk about some of where this is going, that particularly things that involve chemistry. I will try and stay at a high level, but there is actually a little bit of Mike and I had fun talking about enzyme design yesterday. So I thought I'd tell you a little bit about where we are with this. And this really comes back full circle to Jesse's talk, because that was before people even knew it was before there was protein, there was enzyme, or that's not quite right. But before it was known that, well, anyway. So you know what I mean. So now we're actually trying to make enzymes. And so the way we do it is, again, with this diffusion process. So we choose a reaction we want to catalyze. Then we have a substrate or transition state, and then we position catalytic residues around that transition state. And then we run that diffusion process again, and it builds a protein which can hold those residues in just the right place and bind the substrate. So the example that Mike and I were talking about yesterday, this is work by Anna Lalko and Sam, and Anna actually had her PhD defense yesterday on this work. So was to think about one of the very simplest chemical reactions, not simplest, but well-studied chemical reactions, which is ester hydrolysis, and using a serine in a catalytic triad. And I think I have a better slide coming next. So in the catalytic triad, which any of you who've taken basic biochemistry probably remember, you have a serine which is doing the chemistry. It is the nucleophile, and it's activated by a histidine, which is in turn held in place, and there's a lot of this. We could spend several weeks talking about what the aspartate does, but we don't really want to do that. And then there are hydrogen bonds that stabilize what's called the oxyanion at an intermediate step in the reaction. But the point I want to make is that we can actually design, if we position these groups in space, for example, in this version, we just have the histidine and the serine. Here we've added an aspartate or a glutamate, and here we've added an oxyanion hole. And we can now build proteins around these different active sites and see what happens. And so a lot of biochemistry has been looking at the structures of enzymes and making up stories about how they work, because the positions of those groups in space you can't change. You can mutate them, but you can't really move them around, because it took 100, I don't know, a billion years to make trypsin or something. So you can't really experiment in that way. But now we can move these groups around and change what they are and see what happens to catalysis. And so Anna and Sam, and so there's a whole story about this, found that with some arrangements in simple active sites, they could make serines that were nucleophilic, and would react with the probe, but otherwise couldn't do chemistry at all. And then there were other arrangements they found which could do a one round of reaction, but then they would get stuck at an intermediate step in the acyl enzyme. And then there were other arrangements where they got full catalytic turnover. And so this is sort of like a new way of thinking about enzymology. And then structural biology is still important. So this is now taking the, this is now determining the structure of one of the more active designs. And so we can see, just confirm that everything is where we had designed it to be, so that the diffused model is really structurally correct. So one of these, there's two structures imposed here, superimposed here, and one is the crystal structure and the other is the design model. We can do this with other types of reactions. And so this is kind of an easy case. This is also simple ester hydrolysis. This is now using a metal ion, a zinc ion. And this is starting from basically a quantum chemistry, a DFT calculation. So in this case, you can see what the active site looks like. There are histines that coordinate a zinc. And then, I'm not sure if you can see it very well here, but a base that activates the water molecule. And this was very exciting because we made 96 designs, and there was one that was really, really fast, a kcat over km of 22,000, which previously, when we and others had tried to do things like that, this number was more like 10. So this was a big advance. And the other thing that was cool was that its name was A1, because it was actually the top ranked design. So, and the way that we were ranking them was by kind of looking, we developed a deep learning method for sort of trying to sample all the possible states of the active site, both the conformations of those residues that we're trying to hold in place and the substrate or transition state. So this was a bad enzyme that wasn't active, and it was actually predicted, this substrate wasn't predicted to be held together in the right place at all. And this was that really good one. And the reason it was ranked well top was it was really, the enzyme really not only positioned those catalytic groups in space, but also held the substrate in exactly the right place. So, so we can make much, much better things than we could before. And we can, we have an idea of what the important properties are. So there's some very important esters like plastics. Many plastics are esters. So we're trying to use these methods to make enzymes that break down plastic, and other esters that are environmental pollutants. One step harder than breaking down an ester is breaking down an amide. Proteins are made out of amide. The peptide backbone is amide bonds between amino acids. So what we really would like to do, I didn't show you, but I sort of told you that we can make binders pretty easily to any protein we want. Now we want to put catalytic sites into those binders so they actually destroy the protein. And so then you could have a medicine that isn't just blocking a protein, but it's actually destroying the target, like the virus or the bacteria or whatever, which could mean that you might need much less enzyme, much less drug, and it could be much more effective. And we've been talking with Jesse's group about other things to do with metalloenzymes and starting some exciting collaborations there. Just to show you some other examples of reactions, it's now kind of funny after many years where enzyme design just really wasn't working. Here's another case where this came straight from a quantum chemistry calculation. So the click reaction is very widely used in chemistry, and we can now, and it generally requires copper, which is toxic in biomolecular systems, and we can now design enzymes which are quite proficient at catalyzing this. We've actually got good at designing proteins that bind pretty complex clusters like this one. We have a crystal structure now, and this opens up all sorts of opportunities for new metallocatalysts, because these clusters have open coordination sites. So again, very excited about collaborating with people on this. And then I think I will skip through this. This is so nature. Jesse talked about hemoglobin, and which uses hemes, and many enzymes in nature use hemes as well. And we can now design new proteins that have hemes that do some very interesting chemistry. But I wanted to. The last thing I want to talk about, I just want to talk for a couple more minutes, is I mentioned already mineralization, like think about abalone shells, the beautiful shells that are calcium carbonate, somehow protein or biological systems makes them come together, and our teeth are like hydroxyapatite. It's like nature has figured out how to do this amazing thing with minerals. So this is something we've been really fascinated by. And so now that we can design proteins with essentially any property we want, we can design proteins that are complementary to inorganic crystal lattices and build proteins that array functional groups in exactly how we want them. And so this is kind of a neat case with hydroxyapatite, where we built a protein around essentially a hydroxyapatite mini crystal shown here. And so the proteins that we designed are they're shown here, and this is a little bit hard to see, but when we looked in the EM, there were these little dark crystals in there, and when we looked at higher magnification, what we saw looked awfully like what you see partway through tooth formation with these sort of little nanocrystals of highly ordered calcium phosphate. So this is really just at the beginning, and this is being worked on by Tracy Hughes, who really it's really been a lot of fun. So I think this is, so this is like using a natural something that biology uses, but we don't have to be restricted to trying to mineralize with things like calcium phosphate. What if we could organize semiconductors? And so as an example of that, we took zinc oxide, and so we designed proteins that had surfaces that we predicted would facilitate zinc oxide crystallization, and then we built them into these sort of cages here. And the zinc oxide mineralizing surface is supposed to be at the center of these triangles. And then when we made the protein, these cages actually form. This is very similar, this basic idea of making self-assembling systems. I mentioned the one approved design medicine, which is a COVID vaccine. It's based something like this, but now it has parts of the receptor binding domain of the virus on it. Anyway, these don't have anything with vaccines, but they have these interfaces here. And then this is now cryo-EM, and what we see is that inside these axes, there's extra density that looks like it looks like a mixture of zinc ions and water. And so we're really excited that we can now study exactly what's going on here and then use it perhaps to pattern more complex materials. So I just wanted to give you those two examples of things that are happening now. And all this stuff, I didn't talk about hardly any of this, but there's been a lot of, just say that this is a whole thing has been done by many fantastic people over the years. And all of it really none of it really could have been done without all the people who solved all those structures. So anyway, there we go, perfect timing. Yeah,
so
sure.
Chemistry to tradition is students get to ask first questions before old people like me.
Yeah.
I'm a synthetic organic chemist, so I feel a lot of these like protein design, like targeting biological molecules. Do you ever envision like have a completely new protein that targets completely synthetic products, like something like a polystyrene or some sort of like extremely just man-made things?
Oh, absolutely. Yeah. Well, like zinc oxide isn't very common in nature. Yeah, or it's not common in design materials. protein biology, I guess I should say. Yeah, I mean, if you have, it's very straightforward now. If you have a small molecule that you would, I think the only issue, we're trying to make polyethylene glycol binding proteins, for example, now, and that's very flexible and very solvated, so that's a little hard. But really, there's no, these methods can be applied to make things that would bind pretty much any small molecule you want, and then hopefully catalyze pretty much any chemical reaction you would want.
Hi, that was a really great talk. So, I know that in their native conformations, a lot of proteins undergo a conformational change when binding to their substrate or throughout carrying out their function. Are your current computational tools able to account for that or design around that?
Yeah, so actually we can design systems which undergo conformational changes when they bind something. And that's really neat. So we can make proteins that will, say, for example, activate signaling by binding to a receptor. And then we've designed like what's called an allosteric modulator that will bind to that protein and totally shut that off. So we're starting to get really good at designing systems with multiple different states. Yeah, well, it's true. Nature has some really wimpy kind of helpless proteins that need chaperones to really get by. But our proteins are, I mean, we don't need chaperones or any of that silliness. Yeah. So, well, but the true answer is that we almost always make many designs, and some of them just are gunk at the bottom of the test tube, and you don't hear about those. No, they get forgotten quick. So it could be that some of those could be rescued by helpers or chaperones, but for almost every problem, we get plenty of, robust, happily folding proteins. So.
Are there like safety implications to like the possibility of like designing such strong binders, or how is that?
Yeah, well, there's been a lot of discussion. Well, probably all of you heard with AI, people are worried about the dangers of AI, mainly with language models, but that's starting to impact protein design as well. We actually had a workshop here that the White House asked us to put on about a year ago to really take a deep dive into that. And there's one important point about all this stuff is that you can do the computation, but to really bring it in the world, you need to make a synthetic piece of DNA. And so I think really the place to, so the DNA synthesis should probably be monitored. For example, you don't want people making the 1918 Spanish flu or botulinum toxin, which are probably both a lot more dangerous than anything anyone could design right now. But I think there has been a lot of discussion about that. And I think really sort of thinking about how DNA synthesis is monitored is the right way to go. Yeah. Yeah. Well, one place it comes in is we're trying to make, we're designing DNA binding proteins like transcriptional regulators, and they have to be a lot more specific for human cells than for E. coli because there's so much more DNA. As far as like just the stability, they're really pretty similar. I mean, it's around pH 7 or 8 in both. So I think it's more that the complexity of the task, particularly when you want a specific DNA recognition, where it's a harder problem. Yeah, so I mentioned that earlier. Yeah, so we are designing systems now where we design not only the protein, but also the allosteric modulator. Yeah.
It's fascinating to see very real-world results of a computational task. I'm a theorist, so unfortunately, I have to ask a question. When you are going through this process, how can you be sure that once you've assigned these amino acid chains to the structure that you come up with, that you found the global minimum of that energy that you're minimizing?
Yeah, well, you don't know, and that's why, and it's very hard to prove, as you know. You can show that something is not a global minimum, but you can't prove it is a global minimum. And that's why being able to do experiments has been really important all the way along, because, you know, if you make something and it folds to that state, then it's a pretty strong argument.
What are the specific instructions you give in design? Like similar to Dolly Reed described.
Well, it's almost like that. Like if you want to make a binder to a target, you basically provide it the target and say, give it the residue numbers of some residues you want to bind to. You could easily, so there would be an alternative. You could say someone could have a script that then goes to the PDB, gets that structure and indicates those residues. But it's almost as simple as that. So you just, if you want to bind a small molecule, like say something out of that you synthesize, you pretty much just give it that small molecule. Yeah, so it's not quite a text prompt, but it could be. Yeah.
So I was wondering if these methods are able to predict what byproducts will be formed after these proteins are utilized. I was specifically, I guess, one application that I was thinking of was in medications. When proteins are used in medications, there are side effects. And I was wondering if these are
right. So one of the things about, so now that's a really good question. So I talked about designing proteins that do all these different jobs. And we're pretty good now at designing proteins that will carry out a biochemical function. But if you want to make a medicine, then you have to predict what that protein will do in the body. And so if we had public data on clinical trials for many, 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 many different proteins, then maybe one could train a machine learning model to predict what the outcome of a trial would be, or safety or toxicity. But unfortunately, those things are much, much harder, and they aren't really public. So what we can do is do the biochemical things. We can verify that the protein doesn't bind to other proteins in the body. We can verify that it doesn't cause a strong immune response. But you can't really predict exactly what it's going to do, because biology is so complicated. So ML is limited by the data sets you have available. We don't really have the kind of biology data sets you'd need to answer that question. Yeah.
Are there experiments like in this area, like in animal models, like in mice, maybe to like see how it would work like in vivo, and if so, how far along?
Well, I think it's just like one way to think about it is each protein structure, and there are 200,000 protein structures. Each structure has the coordinates of many thousands of atoms, each with an X, Y, and Z coordinate. So the amount of data we have on protein structures, it's like 200,000 protein structures times 1,000 atom coordinates. It's like millions and millions of really, really accurately known data points. So there are mouse model data, but collecting enough data, like millions or billions of data points on mouse experiments is hard. So that's definitely an area of research, but it's just challenging.
David, we've got a couple of questions on Zoom and then maybe one or two more from.
Okay.
Oh, okay. I promise I did not plan this question, even though you've heard me ask it before. But if you have a known protein that can perform the reaction, what's the benefit from designing de novo, or should you stick to enzyme engineering?
Oh, yeah. Let's see. Well, it depends. If it's, I could give a silly answer, I could give a serious answer, I guess.
Both.
Okay. So I'll give the serious answer first. I think it really depends on what you want to do. If it's a reaction that's very similar to one that a naturally occurring enzyme catalyzes, then you're probably better off adapting the enzyme you already have. If it's something that's really different, then you might be better off binding one from scratch. And it reminds me of a story that I think I told Jesse before. I was at a meeting once with Frances Arnold, who actually got the Nobel Prize for enzyme engineering, and she has loved to ridicule protein design over the years. So at the beginning, it was a kind of an audience sort of like this one at a meeting, as she said to the students in the audience, you know, you may hear about people trying to, design proteins on the computers, but it doesn't really work. But the problem she had, the mistake she made, is that I was actually giving a later talk. So I said to the students on the audience, I want you to think back, say Francis and I were back in the Stone Age. And I had this round thing that I was rolling, and I was like, this is pretty cool. And Frances was dragging some heavy thing, and she said, "We don't need that, you know. We got it all figured out." So, so anyway, it probably depends on what exactly you want to do.
And then, okay, sorry, one more quick one from Zoom and then a couple more from the room. So the other Zoom question was, can you do the design with unnatural amino acids?
Yes, you can. I didn't show examples, but you can include unnatural amino acids. So we're doing some chemistry now of photosensitizers, like unnatural amino acids that will absorb light. And so you can put in unnatural cofactors that do more exotic chemistries. So we're collaborating with John Hartwig's group a little bit on that. So yes, the deep learning part, the ML models are a little bit challenged by that because they haven't seen as many examples of the unnatural amino acids, but they're kind of ways you can work around that. Yeah.
That's a bit of a different question. During the process of your research, did you ever have a big eureka moment, or was it more like slow, steady progress?
Let's see. Well, that came up with we went back and looked at it. I think it was kind of just a lot of really smart, energetic people working on stuff over a long time. Really, that's kind of what it was. I mean, with Top 7, I was talking with Brian Coleman and Gautam Dantas, who Brian did the computational work and Gautam did the experimental work, and about what the aha moment was. And what Brian said, the aha moment was for him was basically after he'd left the lab to start his own job at UNC, where he's a professor. There was an email sent around from Barry Stoddard's group with a superposition of the computational model of Top 7 and the crystal structure, and they were identical. So that was kind of an aha moment, but it was like 3 years after the experiments had been done. So, but I would say it's more just been kind of building over time.
Maybe two more.
Yeah. There. Yeah, so we haven't really done the second one we haven't done, but one thing that we find for most designed proteins is that they're much more stable than naturally occurring proteins. So they typically don't melt at 95 degrees, whereas natural proteins are, they're a little wimpy. Like I said, they tend to stop working just above the temperature they were meant to work. But as far as designing proteins to be more active at one temperature than the other, that we haven't really tried to do. Yeah, so we have our first, so we've been trying to apply this diffusion approach to RNA, and we just have our first exciting results. Jesse mentioned Riju Das, who, he has set up like a CASP-like experiment for RNA design, where he then goes and makes millions of RNAs. And Andrew Faber, who's a graduate student in my lab, has been working on RNA design. He made predictions, and Riju found that they were just remarkably good. And Andrew's now made some designs in the lab, and the EM structures look pretty similar. So it's RNA design is just starting to work now. Okay, how about two more right back here? I can't hear. Can you talk a little louder? Oh, because when we're doing design, we don't know the sequence, right? So if you're doing structure prediction, you have the sequence because you got it from, the sequence of a gene. But when you're doing design, you're making up something completely new. And generally, function follows structure. So that's why we have to make a structure first. Yeah. 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 It's a little hard to predict expression levels. In practice, we try and come up with higher throughput streams so you can just test a lot a lot of things, and maybe test things in bacteria or in yeast first before going to mammalian cells. All right, well, I think we should let people who are trapped here go. But yeah, thanks for all the great questions.